Hi everyone. In this chapter, chapter 14, we're going to be talking about allele frequencies. So we've discussed alleles. We know alleles are alternate forms of a gene. And now what we're going to do is we're going to move from talking about individuals and combinations of alleles that individuals have, and we're going to move on to talk about those alleles in populations and some of the things that we can do with looking at these numbers and frequencies. So we'll talk about the importance of allele frequencies in this lecture. We'll talk about what we can do when allele frequencies stay constant. And it's kind of a little bit of a misnomer there, but we will discuss that. And then we're going to talk about an application of this information. We're going to be talking about DNA profiling. So the ability to create a specific, unique profile based on someone's DNA. And there's two different methods for doing this that we're going to actually discuss towards the end of this lecture. All right, so again, we've spent most of our discussion time discussing genetics, talking about, again, individuals. So we learned about genotypes and phenotypes and patterns of inheritance and all of those things that were, again, unique to individuals or perhaps even families. We're going to move that now and go a little bit bigger, and we're going to start talking about how genes behave in populations. So just as a reminder, a population is any member of the same species in a given geographical area who are capable of mating and producing fertile offspring. Okay, so if we're thinking about humans, which for the majority of the course that's kind of what we've been focusing on, you can consider population as small or as big as you want. So there's a population here at Harvard College, a population of students, right? We have a population of people who live in Palatine, Illinois. We have a population of people who live in Illinois. So again, it depends on your given geographical area. And we're talking about just the people right now, but this would also extend to populations of flowers or mushrooms or mice or anything else that we wanted to study. So all of the members of a population share what's called a gene pool. And a gene pool is every single allele in that population. Remember, alleles are alternate forms of a gene. So you're looking at all the different varieties of genes we have in a population. We call that the gene pool. So what we can do with some very simple math is we can figure out how often an allele occurs in a particular population. So the way to do this is we calculate this as the proportion of one allele relative to all of the alleles at that locus, which locus is just a place on a chromosome. So what we're looking at is we want to figure out in a population very specific allele frequencies. So in the previous lectures when we were talking about inheritance of things like cystic fibrosis and sickle cell disease and those types of things, we talked about the frequency, right? So people who with a Caucasian ancestry had a 1 in 300 chance of being a carrier for cystic fibrosis. But how do we know that? Well, we know that by calculating allele frequencies. So this is where this comes into play. If we want to figure out how often the sickle cell allele occurs in a population, and how likely it is that two individuals could come together and pass along sickle cell disease, this is the information that we would use. So again, it's very, very simple to do, and it requires just some very basic math. So let's start here and say that we had 50 people in a population. So it's a relatively small group of people, okay? So we had 20 individuals who were completely normal for hemoglobin. So HB is our hemoglobin allele. And this capital A here just means that it's the normal hemoglobin, okay? So they have normal red blood cells. We have 20 individuals who have two alleles for normal red blood cells. We have 20 individuals who have one normal red blood cell and one sickle-celled hemoglobin blood cells. And then we have 10 individuals who have two of these sickle cell alleles. So all of their alleles, all of their hemoglobin will be, again, is that sickle cell shape rather than those nice round red blood cells. All right, so to figure this out, we take the frequency, we're going to calculate the frequency. It doesn't matter which allele you pick. You can pick either one you want. We're going to pick the frequency of the normal allele, what we're calling the capital A allele. And to do this, we take the sum of all the alleles, A alleles, divided by the total number of alleles. Okay, so let's walk through this. Let's start with our normal individuals first, the ones who have the normal hemoglobin alleles. So there are 40 of those. How do we know? Because we have 20 individuals who have 2 copies of that allele each. So 20 times 2 is 40. And then we're going to add it to the 20 alleles we get from the heterozygotes. Okay, so remember the heterozygotes have 1 copy, so 20 times 1 is 20. So that's going to give us 60. Okay, so we have 60 alleles, right? There's a total of 100 alleles. How do I know this? Because there's 50 individuals. And everybody has 2 alleles, right, for every trait. So you have 2. One you got from mom and one you got from dad. So in order to calculate the total number of alleles in the population, you just multiply the number of individuals by 2. Okay, so 60 divided by 100 is 0.6. So that's the frequency of the regular hemoglobin allele. So what about the sickle cell allele, right? That's what we really want to figure out. So in order to determine the frequency of the sickle cell allele, we can just subtract 0.6 from 1 because the frequencies of alleles will always add up to 1, right? We have to get 100% of the alleles. So if we know 60% of the alleles are the normal hemoglobin alleles, that must mean 1 minus 0.6 is 40%. So the frequency of the sickle cell allele is 40%. Now, we could have started off by just calculating the frequency of the sickle cell allele. We didn't have to calculate the frequency of the normal hemoglobin allele first, but just for illustrative purposes, we did that, okay? If you're confused, don't worry. There is going to be a practice worksheet up on Blackboard that will enable you to practice these calculations. So, again, don't worry about it. Watch this segment a couple times if you need help. Again, I already talked about the total number of alleles is twice the population size. There were 50 individuals. That means there was 100 alleles. Okay, so here's a practice problem. I'm not going to walk through it, but this would be an appropriate point to pause and see if you can work through this problem yourself. So if there's 25 students and we're looking at the frequencies for hair type, curly hair, wavy hair, and straight hair, how many capital big C alleles, curly hair alleles all there, and how many straight or little c alleles are there. Okay, so again, good point to pause here and calculate and see if you can figure out and get these answers. In addition to figuring out allele frequencies, we can also do genotype frequencies. And genotype frequencies are very easy. We just look at the proportion of the population that has one genotype relative to all other genotypes for that particular trait. Okay, so in the previous example, we had 10 individuals who were homozygous for sickle cell out of 50. So that means 20% of the population had the genotype for sickle cell disease. Okay, so that one's real simple. You just look at the number of people you have and you divide it by or the number of, genotypes you have, and just divide it by the total number of people in your population. So that's a pretty simple one. Again, here's another practice problem for you. So going back to the students, looking at the types of hair that they have. So what's the frequency of curly, wavy, and straight hair? Okay, really simple. You take the number of students who have that genotype and divide it by the total number of students. So this is a really easy calculation, but one that you should also be able to do. Okay, so great. We can calculate allele frequencies. So why is this important? Again, one of the things that we can do is we can determine how frequent disease-causing alleles are for simple Mendelian traits. So if we're looking at sickle cell and cystic fibrosis and Tay-Sachs and achondroplasia and some of those single gene traits that we've already discussed, we can look at the risk of inheriting a genetic condition. So if we know that in this population, the frequency of the sickle cell allele is 20%, then we can sort of start to calculate what the risks are of inheriting certain genetic conditions. So this sort of ties into the idea that in order to make these calculations, in order to determine these frequencies and these allele frequencies, we are basically assuming that the allele frequencies never change, that the allele frequencies are constant in a population. And if 20% of the population has the sickle cell allele, then 20% of the population will always have the sickle cell allele. And with that, you can do some sort of fancy math. And that fancy math, and I'm not going to go into it too much, is called the Hardy-Weinberg equilibrium. And what Hardy-Weinberg equilibrium means is that we can predict offspring phenotypes. So we can predict what's going to happen in the next generation by looking at the frequencies of alleles in the parent population. So this is interesting, to say the least, and there's some, again, kind of fancy math that we can do. But the problem with Hardy-Weinberg is allele frequencies aren't constant in populations. That's what we're going to talk about in our next lecture in chapter 15. This isn't really reality. Allele frequencies are constantly shifting and they're constantly changing, and they're doing that for a variety of reasons. And so you can't always assume that those allele frequencies are going to stay the same from generation to generation. So it's very rare for these protein encoding genes that sort of influence, again, our traits. We can't really use it for that. But what we can use it for is something else. It's actually useful for the non-coding regions of our DNA. So the non-coding regions of our DNA pretty much stay the same from generation to generation. And the reason is because we don't have what are called selective pressures. And again, I'm kind of leading myself into my next lecture, which is all going to be about selection. So don't worry about that. But what it means is that there's no reason for the non-coding regions to change from one generation to the next. We use this information as a basis for creating a DNA profile. If you didn't know, a DNA profile is a method of identifying an individual by unique characteristics of your DNA. So it's not your entire sequence of DNA. That's not what we're looking at. So we're not laying out the sequence of all of your bases from beginning to end. That would be extremely costly and kind of a waste of information. We don't need all that information. All we need to do is we need to find some characteristics that everyone shares in their non-coding region, and we need to see how they compare. And that's how we can create a DNA profile. So your DNA profile is not based on your protein encoding genes. It's got nothing to do with the traits that you possess per se. What it has to do with it has to do with the non-coding regions of DNA. So those places in between genes. And those places in between genes have a lot of variability amongst individuals. So we can look at two different things when we're creating a profile. We can use either something called short tandem repeats, which are shortened by STRs, or restriction fragment length polymorphisms, which are known as RFLIPs for short. So I'm going to go through both types of analysis to show you how we create a DNA profile. We're going to start with RFLIP analysis. And RFLIP analysis was kind of the very first DNA profile that was created. Basically what you do is you take a sample of from someone, again, it has to have their cells, it has to contain cells, and you break the cells down to release the DNA. If we have a very, very small amount of DNA, we can actually make copies of it with a technology known as PCR, which is called polymerase chain reaction, but don't read too much into that. The idea here is that we only need a teeny, teeny, tiny sample, and we can get the DNA that we need. So next what we do is we take our DNA that we've isolated from those cells, and we cut it into pieces using something called restriction enzymes. So these restriction enzymes are enzymes that will cut the DNA at a very specific sequence for every single person. So in every single person's DNA, there's a sequence there that we're looking for, and we cut the DNA exactly at that sequence. Okay, so the enzyme would sort of cut this piece and cut it into two fragments is what we're looking for. So next, these fragments are called what we call restriction fragments, and you get thousands of restriction fragments of all different sizes because that sequence that we were looking for may be many, many, many bases apart in some people, which creates a really long piece of DNA, or that sequence that we're looking for could be really close together to the next sequence, and so it's a very short fragment. So what's different in every person is the size of your fragments. Okay? Lots of little fragments, lots of big fragments, something in between. So we take all of those fragments and we use something called gel electrophoresis. Okay, so here's what a gel chamber would look like. And we have this agarose gel. It's kind of like clear jello is the best way to think of it. And we take your fragments, your DNA fragments, and we put them and load them up into our gel. And we use some electricity. Okay, so as it turns out, DNA is actually attracted to that electricity. So DNA is negatively charged, and it's attracted to the positive charge of the electricity. So these fragments of DNA are going to begin to move towards the electricity. And the smaller the fragments are, the farther they travel. And the larger the fragments are, they stay at the top. So what you get is this unique banding pattern that is unique to you and nobody else, unless you have an identical twin, and then you'll have the same banding pattern. But assuming you don't have an identical twin you don't know about, your banding pattern will be different, and this is your DNA profile. This is what makes you unique and special and different than everyone else. Then what we do is we basically use a radioactive material and makes a fluorescent image, and we take a picture. So what you're looking at here are actual DNA profiles of individuals. Okay, and again, some of them are the same because we loaded the same sample, but a lot of times what we'll do is we'll use this for identification purposes, or let's say it's like a paternity suit, and we've got we want to know who's the father of a child. So we would put the child's DNA in here, and then we would load the sample potential fathers in here, and we would look for pattern matching is what we would do. However, quick note, rig flip analysis is old. It's old technology. We don't use it anymore. So anytime you're watching your favorite crime scene drama and they talk about a DNA profile, it wasn't created using rig flip analysis. It was created using short tandem repeats, or STRs. So STRs is something different. We are specifically looking at, once again, these repeat regions, but rather than cutting apart our DNA into pieces, we're going to just count the repeats themselves. So we're looking for a specific sequence and how many times it repeats. And based on the number of times it repeats, it gives you a number, okay? A number of repeats at that particular location on your chromosomes. So just like with alleles, homozygotes and heterozygotes, if you have two chromosomes with the same number of repeats, you would be homozygous for that repeat allele. If you have one of each, you would be heterozygous. So we use some of the same technology. But basically, if you wanted to look and see what this looks like, so here is a region of human chromosome number 7. So this was obtained from GenBank, which is a public DNA database. And what we're looking for is the sequence GATA. So GATA, 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 over and over and over and over and over again. That's what we're looking for. So the number of times it repeats is your number. So this, again, is kind of largely replaced rig flip analysis, and this is what we're now using to create DNA profiles. So we load it into a machine, and the machine actually counts the number of repeats. That's basically the way that it works. So I'm kind of going through this quickly, but don't worry about all the individual steps. So here's where we put our samples, and here's where the sample goes in, and this is the big fancy machine that reads it. And when you're done, you get a digital output which has little peaks, which tells us how many short tandem repeats you have. So let's take a look at this. This is what a profile would look like. Okay, so these are for the different locations that we tested, okay, for the number of repeats, okay? And again, we're looking at the peaks here again. So this is for location 1, location 2, 3, 4, so on and so forth, right? And if you only see one peak, that means they were homozygous. They had two chromosomes that had the same number of repeats. For all of the others, we're looking at different repeats, right? So this is your genetic profile, all right? So this is your unique identity as far as your DNA is concerned. So again, we don't create profiles based on what genes you have, what traits you have. We create a profile based on the number of repeats that you have that are unique to your DNA. So what can we use it for? All kinds of things. Forensic work on crime scenes, testing parentage, identifying victims of mass disasters. We can use it for animal identification, looking at racehorses. We can use it for conservation biology. We can use it for a whole host of things. So probably the biggest field for DNA profiling is forensics. And in forensics, we're using scientific knowledge in legal situations. So what we do is we create these DNA profiles to be able to place a person at the scene of something that happened. So the chances of two people having exactly the same DNA profile is 30,000 million to 1, except for identical twins. So what that means is there's basically no chance that you share a profile with someone coincidentally. Anything that contains your cells, we can get a sample from: blood, hair, saliva, semen, body tissue. We have gotten samples from all kinds of things. And again, we can use it to help us solve crimes. So we can match a DNA that's found at the scene of a crime with the DNA of a suspect or a person of interest. It doesn't prove that they committed a crime. It just proves that they were at the scene of the crime. So we can place someone there because of their DNA. Okay, so important terms and concepts to know for Chapter 14.